Okay, so we are now going into proteins. We have finished talking about lipids, cholesterol, and fats, and we talked about how the lipids are important for the structural integrity of the cell because 98% of the cell membrane is made up of lipids, but they are a special type of lipid known as phospholipids. And the most important thing we have to know is that water is very important for joining together these micromolecules and turning them into macromolecules. But if we want to join them, we remove water. We remove water. That's dehydration. You see here, an OH comes from the glycerol, and an H comes from the free fatty acid, and now they are joined together by the removal of water. And if I want to split, I add OH to one side, and I add an H to the other side, and now I have split them. So that is the main difference. So we see that this is happening not only with fats. Remember, this happened also with sugars. You see these sugars, you see this O bond? It's also made by the removal of water and by the addition of water. So that shows you the significance of water. And it's a chemical reaction where water can join together micromolecules by the removal of water minus H2O, and water could be added into the macromolecule to separate it into two pieces. Correct? Are we through with that? Do you now appreciate how water could be added to separate macromolecules and how water could be removed to join together macromolecules? Is that point clear? Okay. Now, we're moving on to proteins. We will see the same theme happening again, and that is if we want to join. So all these macromolecules, they are made up of micromolecules. So the sugars are made up of disaccharides and monosaccharides. The lipids are made up of free fatty acids and glycerol, as well as your nucleotides. We will see them when we get to them. And now this is the protein. So now the proteins, macromolecules, are different than the other macromolecules structurally. So that means they will have different functions. So the protein is a polymer of amino acids, and amino acids have a central carbon with 3 attachments. The central carbon has 3 attachments. One of them is a hydrogen bond, and one is the amino acids have a central carbon with 4 attachments, not 3. One is a hydrogen bond. The second one is amino group. The third one is a carboxyl group, and the fourth one is a radical group. So these 20 amino acids are used to make the proteins. They are used to make the proteins. 20 amino acids used to make the proteins, and they have properties for each amino acid determined by the R group. So here, these are the amino acids and the peptides. So now you can tell right now that this is an amino acid. Can you identify the alpha carbon? You are responsible of knowing what structure is this. Maybe you don't need to know, is this methionine? Is this cysteine? Is this tyrosine? Is this arginine? You don't have to know that. You don't have to know which one. But you definitely need to know the structure from far. Is this a lipid? Is this a fatty acid? Is this a sugar? Is this a nucleotide? So you need to know that this is an amino acid. You don't need to know its name. You don't need to know exactly what is it, but you need to know that generally this is an amino acid. This is an amino group. That is an H group. That is a carboxylic group, and that is a R. That's a R group. So you should be able to tell which one is your amino group. It has N with 2 H's. Which one is a carboxylic group? which one is a hydrogen group, and that is your R group. Okay, so a peptide is composed of two or more amino acids joined by peptide bonds. So the peptide bond joins the amino group of one amino acid to the carboxylic group. So you see this OH right here from the carboxylic group. Can you see the OH from the carboxylic group? That OH here and the H from this amino group are going to be removed. And then you're going to get a peptide bond. So please remember that we use water, dehydration, we remove water, minus H2O, so we can tie them together with a peptide bond. So how do we get the water? We get the OH from the carboxylic terminal, and we get the H from the amino terminal. Then they connect. Okay? That point is very important. So the bond that connect two or more amino acids is known as a peptide bond. So peptide bonds join the amino group of one amino acid to the carboxylic group of the other, and they are formed by dehydration synthesis. So we said that dehydration, removal of water, dehydration synthesis. Okay, so a peptide is composed of two or more amino acids joined by a peptide bond. So the peptide bond joins the amino group of one amino acid to the carboxyl group of the next. They are formed by dehydration. You see here, that's the dehydration. That is the dehydration. Dehydration, removal of water. So one side gives OH, the other side gives an H. But that was for lipids and fats. We're going to see the same thing happening, not on lipids and fats, but actually on proteins. You see the OH here and the H from the amino terminal are going to be removed, and then both bonds, this carbon and that nitrogen are going to be connected to have a peptide bond. You see exactly here. So that's amino acid 1. It's a random amino acid. It has a hydrogen. Amino acid number 2 has a hydrogen as well. Then there's an amino terminal. Then there is another amino terminal. Then there is a carboxylic terminal. And then there's the R group. So when we want to join, you need to know that we join by the OH from the carboxyl side of one amino acid and one H from the amino side. So the amino side gives me one H and the carboxylic side gives me OH, and then water is removed. That's called dehydration, condensation. So now they're going to condense and give me this bond right here known as peptide bond. How many amino acids in the peptide bond? There's 2 of them. This is known as a dipeptide bond. Are we clear with that? This is really important. The second thing we need to know is that proteins have a complex 3-dimensional shape, referred to as the conformation. So each protein is different. Some proteins are protein channels that allow for the passage of ions through the they are transmembrane proteins. Some proteins, they are enzymes that phosphorylate other enzymes and activate them. Some other enzymes, they are catalytic enzymes. But in the end, if you are an enzyme, you are a protein. But because these enzymes have different functions, since the enzymes have different functions, that means they will have different structure. So proteins can reversibly change the conformation to affect the function. So important examples are in muscle contraction. We will get to that when we get to muscle contraction. And then there's enzyme catalysis, membrane channel opening, and so on. For denaturation, there's extreme conformational change that destroys the function. So denaturation is extreme conformational change that destroys the function. So extreme heat or pH may cause permanent irreversible denaturation. So an example would be a cooked egg becomes opaque and stiff. That's denaturation. So you need to know what conformation is and what denaturation is. Denaturation is when we heat up a molecule, and that heat is going to destroy the hydrogen bonds. This is really important. Let me see if I can get the camera to be on my. I want to draw this on the board, but I want also. Okay. Okay. So we talked in the past about how many different types of bonds do we have. We said that the bond that's between sodium and chloride, the bond between sodium and chloride, sodium gave an electron to chloride, so chloride is minus, and sodium lost an electron pair, so that makes sodium positive. Okay, so now what kind of bond is that? Are they sharing the electrons? Are they sharing the electron? No, they're not sharing the electron. Sodium gave up its electron to chloride. Like when the husband gives the wife the ring, he doesn't have it anymore, she does, and then they have a bond. But if we look at carbon dioxide, in carbon dioxide, they are sharing the electron. 50% the electrons are with oxygen, 50% with carbon, 50% with carbon, and 50% with oxygen. Okay? You guys see that? So now they are sharing equally. But here, there's no sharing. Here, that's ionic bond. That's an ionic bond. And this sharing here, that shows a covalent bond. And since the sharing is equal, that is known as non-polar covalent bond. But when you look at water where you have really big oxygen binding with really tiny small hydrogen, what's going to happen here is, are they sharing electrons? In water molecule, are the oxygen hydrogen sharing electrons? Yes, they are sharing them. But are the electrons spending the time 50-50 like how it is with carbon and oxygen and carbon dioxide? Of course no. So hydrogen will have 10% sharing and 90% will be with oxygen. You see now? So that's an example where there is sharing, but it's not 50-50. That is known as polar covalent bond. That is a polar covalent bond. This polar covalent bond shows that around one side of the bond, the electrons are polarizing to one side over the other side. Okay? Yes, no, this one is a polar covalent bond because the electrons are polarizing to one side. When it's 50-50, when they are shared, is non-polar because it's not polarizing anywhere. Here, that is non-polar. But here is polar. That's polar covalent bond. Okay, so now the electrons are more around oxygen than they are around hydrogen. Unequal sharing, polarization. They are polarizing. So when the husband and wife have children living in the same house, they have equal sharing. But when they divorce, they spend 80% of the mom and then 20% with the dad. That's an example of polar because the children are more polarized with the mother because she's the primary caregiver than the father. But when they are in the same household, it's 50-50. So when the electrons are being shared, it's 50-50. That's non-polar covalent bond. But when they are shared, that's polar covalent bond. We said this last week. We went over all this last week, so I don't know why. Why are we having trouble remembering that? I don't understand why is it a problem remembering this. So what I want you to understand is that when we look at water, water in a solution looks like that. O with two H's, O with two H's, and O with two H's, and O with two H's. And we know that since this bond here, I think you guys, if you look up here, it's better than, can you see guys well there? Okay, if you look at here, you will see that oxygen, since it's a polar bond, and the electrons are more polarized around the oxygen than they are around the hydrogen, that makes oxygen really electronegative and hydrogen really electropositive. Okay? And oxygen really electronegative and hydrogen really electropositive. Hydrogen really electropositive and oxygen really electronegative. So this is the bond that's inside. But the problem is what's happening is this bond will never remain like that forever because this electronegative this electronegative oxygen is going to say to the electropositive hydrogen, let's have a dotted yellow bond. There is a dotted yellow bond. There is a dotted yellow bond. There is a dotted yellow bond. So this dotted yellow bond is between whom? Between the oxygen, who is really electronegative, and the hydrogen, who is really electropositive. And this oxygen who is really electronegative, and this oxygen who is really, or hydrogen who is really electropositive, they are going, this is electropositive, they are going to bond together, right? But the bond is a yellow dotted weak bond. So the red bond, what is that bond called? What kind of bond is the red bond? Inside the water molecule. What kind of bond is that? What's the name of this bond? We just said it. I'm sorry? I need to hear an answer. What's the name of this red bond inside the water? H2O. This red bond, what's the name of the bond? Is it a peptide bond? Is this connecting two amino acids? Is it what? It's a covalent bond. Is it polar or non-polar? It's a polar covalent bond. Because why are they polar? Why are they covalent? I'm sorry. If you want to tell, they're covalent because they're sharing electrons. So here, between hydrogen and oxygen, are they sharing electrons? Are they sharing electrons? No, because the electron of the hydrogen is 90% with oxygen. If hydrogen cannot share electron with oxygen and share electron, it only has one electron. So hydrogen is already occupied with oxygen, and even with oxygen, its one electron is even pulled more toward oxygen. So what's this bond called? This is not a bond based on electron sharing. This is not a bond based on electron sharing. And don't tell me ionic, because in ionic, somebody already lost an electron. So hydrogen and oxygen here, nobody lost nothing. So please see that these are three different water molecules. Four different. One, that's the H2O. Please look here. That's an H2O. That's an H2O. That's an H2O. That's an H2O. These are four water molecules. So the bond inside, that's known as intramolecular bond. It's inside the bond, inside the molecule. That's why it's called intramolecular bond. But if it's between this water molecule and an adjacent water molecule, so now they're going to have the yellow bond. So the yellow bond is not inside. The yellow bond is between two different separate molecules. So the yellow one is not intra. This is intra. This is intramolecular bond. This red one. But this one here, that's an intermolecular because it's between two. That's an intermolecular bond. So this bond that's red, that's an intramolecular bond. That's a covalent. That's a covalent intramolecular bond. Okay? But here, that's an intermolecular bond that is based on, what's the name of the bond? The yellow dotted bond between, you can look at who is it between. Oxygen and hydrogen, that's intermolecular. But what is it? Is it covalent? Covalent, intermolecular. It's not covalent because they're not sharing electron. What is the law? What is the rule for covalent? You have to share electron. But this oxygen is sharing with this, and this hydrogen is sharing with that. That's covalent. But then those two, intra is covalent. Intra is covalent. But between the two, intermolecular is not covalent. They're not sharing electron. Is it ionic? It's not ionic because one is not losing electron. No one is oxidized. No one is reduced. Then what is this bond? Oh, it's on the slides. You guys didn't go back, read the slides. The last time you dealt with this course was when you saw me last week. Okay, that's not how things work. This is a hydrogen bond. This is a hydrogen bond. So because it's between the really electropositive hydrogen and the really electronegative oxygen. So now that's a hydrogen bond. This hydrogen bonding is so important because that is how your protein is going to flip on itself. It's going to have a conformational change. So now it's going to move from a primary protein to a secondary protein to a tertiary protein to a quaternary protein. These are globular proteins. These are proteins that flip up on each other. Okay, these are proteins that are globular, that flip on the top of each other. Okay, so the problem now is when we heat up a globular protein that is quaternary protein, tertiary protein, secondary proteins. When we heat them up, what causes them to go from quaternary to tertiary to secondary to primary? It's the loss of the hydrogen bond, because who made them? What made the primary protein sequence become secondary? It is the hydrogen bonding. What made the secondary protein conformation become tertiary protein conformation is more extensive hydrogen bonding. What made the tertiary protein becomes a quaternary is more extensive hydrogen bonding. So all of this conformational folding, when it folds upon itself from primary to secondary, secondary to tertiary, tertiary to quaternary, these are flipping up on themselves. So they are flipping up on themselves due to hydrogen bonding, not covalent bonding. So when I heat up the protein, it's going to flip back. it's gonna go back to third. The more I heat, it's gonna go back to second. The more I heat, it's gonna go back to first structure. But when I cool it back up, it's gonna return again to the secondary, then tertiary, then quaternary. Do you understand? This hydrogen yellow dotted bond is the reason why all your macromod in the DNA, why the DNA look like that, double helix. It's a double helix because the bond that is holding the two templates together is a hydrogen bond. So when you heat up the DNA, it's going to fall into two pieces because the hydrogen bonds are broken. When you cool it again and heat leaves, they're going to now reform. So that's why we have to appreciate this yellow dotted bond, this hydrogen bond, because it is behind all the conformational folding that takes place. Clear? Does anybody have a question? Okay, now let's go back to our slides. Okay, so this is so now we said that the protein structure, proteins have so that's a sequence. What we see right here, amino acid 1 attaching to amino acid 2 horizontally, like the train. You know the trains are connected by a bond. So think of the amino acid as train cars. So they connect to one another. They connect to one another by bonds. So what's the name of the bond that connects all the amino acids together? What's the name of the bond that connect all of the amino acids together? Peptide bonds. So now proteins have a complex, proteins have a complex three-dimensional shape referred to as the conformational shape. And that is unique and crucial to the function of the protein. So proteins can reversibly change the conformation to affect the function. So important examples seen in muscle contraction, enzyme catalysis, membrane channel opening, and so on. So denaturation is extreme conformational change that destroys the function. That's extreme conformational change that destroys the function. So the extreme heat or pH may cause permanent irreversible denaturation. So when you heat things beyond their capacity, so now you cannot have this reversible. Remember when I mentioned how when you heat up the DNA, it's going to denature. When I cool it, it's going to return back again. But if you heat it too much, you're going to destroy the bonds. Now you even destroy the covalent bonds. So now even if you cool it, that is known as damage beyond salvage. Damage beyond salvage. Okay. Proteins have 3 to 4 levels of complexity. There is the primary sequence, the primary structure, and the sequence of amino acids within the protein molecule. So the primary structure is encoded by the genes. So a secondary structure is coiled or folded shape held together by hydrogen bonds. So the hydrogen bonds between slightly negative carbonyl and a slightly positive NH group. This is so important. So now you're going to see how the hydrogen bonding is going to happen. So now we know, does primary structure have hydrogen bonding? Does primary structure have hydrogen bonding? No, because they are like a train. They are connected to each other only with peptide bonding. But then this peptide bond is going to flip on the head of itself. And who's going to connect? The hydrogen of this nitrogen to the O of this carbonyl. That's how they're going to make the change. You see, it's right here. It's coiled. The secondary is the first coil or folded shape held together by hydrogen bond. The hydrogen bond is between who? It's between the slightly negative oxygen of the carbonyl bond and slightly positive NH groups. So the most common secondary structures are the alpha helix, they are like spring-like shape, and the beta sheet, they are beta pleated sheets. So these two, that's a perfect example of a secondary protein structure, the alpha helix and the beta sheet, the alpha helix and the beta sheet, beta pleated sheet. So if you're asked on the exam, alpha helix and beta pleated sheet, are they primary structure, secondary structure, tertiary structure, and quaternary structure? Secondary structure. Protein levels of complexity. Continued. The tertiary structure is known as further bending and folding of proteins into globular and fibrous shapes due to hydrophobic interactions and van der Waals. So now we have, in addition to hydrogen bonding, you have additional hydrophobic interactions and van der Waals, and that gives us a tertiary structure. What's the perfect example of a tertiary structure protein? Myoglobin. Myoglobin. That's a third tertiary structure protein. So now we're talking about the different types of bonds that enable us to have a tertiary structure. So we have the hydrophobic interactions, we have the van der Waals forces, and we have the disulfide bridges. And the disulfide bridges are between the cysteine amino acids that stabilize the tertiary structure. The cysteine amino acids that stabilize the tertiary structure. There are globular proteins. They are globular proteins, and these globular proteins are compact tertiary structure for proteins within the cell membrane and proteins that move freely in body fluids. So you see, there's a compact tertiary structure for proteins within the cell, and then there are fibrous proteins. These are slender filaments suited for roles in muscle contraction and strengthening of skin and hair. Okay, protein levels of complexity. There is the quaternary structure. Quaternary structures are associations of two or more polypeptide chains due to ionic bonds and hydrophobic interactions, and they occur only in some proteins. So for example, hemoglobin has 4 peptide subunits. So that's a perfect example of a quaternary structure. So, and that happens when you have an association of 2 or more polypeptide chains due to ionic bonds and hydrophobic interactions. Now, let's look at pictures. These are perfect illustrations. Whenever you're studying for your exam, you really need to make sure that you haven't left any picture out, any figure. You see these figures, you have to know all of them because they are all part of your lecture questions and lab questions. These are key concepts. So maybe on the exam, I'm not going to bring this particular exact figure, but the concept, you will be tested on it. I promise you. So let's look at the primary structure. Remember I said the primary structure is like what? It's like the train track. They are like the train cars connected to each other. So that's a yellow amino acid, purple amino acid. What's the name of this bond connecting all of these amino acids? That's a peptide bond. And that shows you they are like a string of beads. They are all connected horizontally. Nothing happening, no folding. So the primary structure is the sequence of the amino acid that is joined by peptide bonds. And that is the most important ones because when you heat up the protein, it falls back into its primary structure. When you cool it again, it returns back into its quaternary structure. That shows you the most important structure is the primary structure. Then you have secondary structure. The secondary structure is the alpha helix or beta sheet formed by hydrogen bonding. So the secondary structure, alpha helix or beta sheet, formed by hydrogen bonding. Tertiary structure are a single polypeptide chain folded and coiled by interactions among the R groups and between R groups and surrounding water. That's the tertiary structure. A single polypeptide chain folded and coiled between R. So now the R groups surrounding the water are going to have hydrophobic and van der Waals interactions. That's what causes them to flip. Then we have quaternary structure, and these are associations of two or more polypeptide chains. So here you have polypeptide chains 1, 2, 3, 4, and they are all associated by ionic bonds, and they are associated by hydrophobic interactions. And that, you see how we're flipping? We flip from primary into secondary, then we flip from secondary into tertiary, then we flip from tertiary into quaternary, and that in the end gave us our main target. That is our main target protein. But we use different forces. We use different forces to enable us to fold. So when we folded from primary to secondary, that was hydrogen bonding. When we flipped from secondary to tertiary, that was hydrophobic interactions and van der Waals interactions between the R groups. And then when we flipped from tertiary to quaternary, then that helped us develop ionic bonds and help us develop more hydrophobic bonds and hydrogen bonds, and that led us to have a quaternary structure. So these figures are important because they describe to you. Now you're going to have a mental picture. So always go to the figures, they help you a lot. So the protein structure, conjugated proteins contain a non-amino acid moiety. Moiety is an addition, known as the prosthetic group. The prosthetic group are covalently linked to the protein. So for example, we said hemoglobin is a protein that has a prosthetic group known as a heme group or a heme moiety, and that heme group and the heme moiety binds it has an iron and it binds oxygen. It binds oxygen. So proteins have more diverse functions than other macromolecules. So for example, remember we talked in the past about structural proteins and functional proteins. So structural proteins like keratin, that is a tough structural protein of the hair, the nails, and on the skin surface. And the collagen is contained in the deeper layer of the skin, and that is for your bones, your cartilage, and for your teeth. And proteins are important not just for structure, they're also important for function, such as transport, like for example, hemoglobin transports oxygen. That's not there for structure. That's there for the function of providing oxygen to the tissue that needs oxygen and removing carbon dioxide from tissue that needs carbon dioxide to be removed. So remember, hemoglobin has a downtown trip and an uptown trip. Hemoglobin has a downtown trip from the lung to the tissue to pick up oxygen and deliver it to the tissue that is exhausted, a tissue that is blue because oxidative phosphorylation and aerobic respiration are happening, consuming all the oxygen. So then hemoglobin will let go. It will have low affinity for oxygen and let go of the oxygen in the tissue. Then we have an uphill trip where hemoglobin pick up carbon dioxide and goes and deposit it into your lung so the lung can get rid of it. Another type of the importance of, now we are appreciating the different important uses of your proteins. They are there for structure. An example of that is keratin and collagen. An example of the functional protein is your hemoglobin. And an example of communication is the neurotransmitters. So neurotransmitters are some hormones and other signaling molecules and proteins. So signaling molecules, that exert their effects by reversibly binding to a receptor molecule are called ligands. Ligands. So a ligand would bind, it's a messenger that would bind to the receptor, and once it binds the receptor, that would lead to the opening of the protein channel. So what is a ligand? What is a ligand? So we have transmembrane proteins. These transmembrane proteins are very important. These transmembrane proteins are very important because we said that the cell membrane looks like this. It has a phospholipid bilayer. A phospholipid So the plasma membrane is made up of two leaflets, an upper leaflet and a lower leaflet. This leaflet, when we studied the fats, remember, when we studied the fats, we said that we have a phospholipid. The phospholipid has a phosphate group that is charged, and it has a choline group, and then you have your lipid tail. So that shows you this is the polar head of the phospholipid, and this is the non-polar tail. So these are the tails and these are the heads. So if I have a hormone that is made of cholesterol, a hormone made of cholesterol, a hormone made of lipids like aldosterone, testosterone, progesterone. These hormones are welcome. They can cross the lipid bilayer because they are also made up of lipids. Remember we said like dissolve like. The polars dissolve polar. Salt is dissolved in water because salt is polar. One has a salt, sodium has positive charge, chloride has negative charge. Sugars are polar. Glucose is polar. That's why it dissolves in water. Now try dissolving glucose in oil or dissolving salt in oil. It's not going to dissolve because they are made up of different substances. But if you try to dissolve soap in oil, it will dissolve. Soap in detergent will dissolve. Detergent in oil will dissolve. So here, that steroid hormone, aldosterone, progesterone, it can dissolve in the lipid bilayer tail. You see this? The core is hydrophobic, lipophilic. This is butter. This is yellow butter. This is lipid. So if you are lipid, you can cross easy, and they have receptors that are found in the cytoplasm, or they can even cross the nucleus. They can go inside where there is the DNA and transcribe for us after they bind to DNA-bound receptors and cause their transcription. Okay? Does it make sense? Okay. Okay. All right, guys. Now, if you are non-polar, you cannot enter. So now we need a channel to get you in. We need a channel to open up to get you in. Some channels are based on a ligand. So the ligand would bind. When the ligand binds, the channel would open, and then you would have entry known as influx, or they would exit known as efflux. So this is what a ligand is. A ligand has to bind like a lock and a key. So one ligand looks like that, it will only bind this. One ligand looks like that, it will only bind this. One ligand looks like that, it will only bind this. So the ligand binds with a conformational specificity. So it's like a lock and a key. You cannot open that door unless you have the exact key for that. So now, some proteins are ligands. These are ligands. And the receptor who's accepting them is a protein too. And the channel that opens up to allow them to pass is a protein too. And the enzyme who gives me my target effect in the cell is a protein. The transcription factor, protein. Proteins are what run our life. That's why the central dogma of biology is to transcribe the DNA to get you mRNA and to translate the mRNA to give you proteins.
The ligand, it binds with a protein that's similar to it.
Yeah, yeah. And then that causes an effect. They open to allow the entry of something. Okay. You see, there we are. Now, membrane protein functions. We talked about one function. They are structural, like keratin and collagen. They are important for communication, like ligand. They communicate between two cells. And there's also membrane transport. These are channels that allow hydrophobic substances to diffuse across the cell membrane. And there are carriers that help the solute cross cell membranes via active or passive transport. There is also catalysis. So in catalysis, the enzymes that catalyze physiological reactions are usually globular proteins. These are enzymes. We talked about enzymes last week, how enzymes are very important because they help us. What do enzymes do? Yeah, how do enzymes speed up the reaction, the chemical reaction? Yeah, so how do they do that? If I have a reaction coordinate and I have reactants here, and I have product here, and I need to go from reactant into product. So I need to have an activation energy barrier, EA. What happens now? What does the enzyme do in order to speed up the reaction? This is Mount Everest. We need to climb this mountain to go from reactant A to reactant B. Okay? But the problem is, this is taking a long time. So an enzyme would come and say, you don't have to take this route, number one, to go from A to B. I have a shortcut. I get you there faster. So what does it do? I said this last week. So what does it accomplish? It lowered what? It lowered EA. So now you don't need to go up this much. You only need to go up this much. So we take this route B, this route number 2, because EA now went smaller. EA of reaction 1 is smaller than EA of reaction 2. So they lower the activation energy barrier. And that's what makes the reaction go faster. Oh, wow. Oh, boy. Oh, no. Recognition and protection. I wanted to give you freedom, a little bit of homework so you can go. I don't want my homeworks to bother you and stop you from freely. That's how I used to be when I was in college. I don't want to be bothered with assignments. I want to read and learn at my own pace. But now, obviously, this is not happening. Obviously, this is not happening. We are repeating what happened last week, and the people are not remembering. So please go home and read. Don't expect you see me once a week and then that's it. You don't need to deal with this course again until you see me again next Thursday. Recognition and protection. So glycoproteins are important for immune recognition. Antibodies are proteins. They recognize, antibodies recognize, and they are able to, so antibodies are proteins that recognize glycoproteins. So sometimes proteins have an association with a sugar. on the surface of our cells. We have a sugar on the surface of our cells, and then our immune system is screening that glycoprotein. We will learn that today with the glycocalyx. Proteins are also important for movement. So they are molecular motors, molecules with the ability to change the shape and repeatedly, like albumin. That helps, is like a carrier in the blood. It helps transport, you know, the LDL, we're talking about LDL. LDL, what's the difference between LDL and HDL? What's the difference between low density lipoproteins and high density lipoproteins? We said that last week. Yes. Can somebody please tell me a legible answer, a complete sentence, so I can see what I can correct, what I can. I don't want mumbling. What's the difference between LDL and HDL? Yeah. HDL is the good cholesterol, LDL is the bad cholesterol. But what makes it bad? What makes it good? Yeah, so what about the density? Excellent. What about the density? Excellent. I'm sorry? Yeah, so from the name, low density lipoprotein, high density lipoprotein. Okay, both of them, LDL and HDL, HDL and LDL. In HDL, you have a lot of protein, that's the carrier, that's the boat, and you have a little bit of fat in the center. So you have a high ratio of protein, who's carrier, and a low ratio of fat. That's high HDL. But here, you have somebody who eats so much lipid, so much cholesterol, so much fat. So when you look inside, the carrier is not like that one. The carrier, a lot of protein, a little bit of fat. Here you have the protein, you have a lot of fat inside. So here you have more fat and less protein, and that is low density. So that's what makes LDL bad for the heart, for cholesterol. You understand? Do you understand?
Yeah, less protein.
Yeah. So if you have more fat, less protein, that makes it hot, that makes it low density LDL. That's why they call LDL bad cholesterol. So LDL is bad cholesterol. It has a lot of LDL is bad cholesterol. It has a lot of protein and it has a little bit of protein and a lot of the ratio for lipid to protein is so low. And then that shows you there is low protein and there is high fat and lipid. Now let's look at here. What do we have here? Here we have a lot of protein and a little bit of fat. So that is high density. So if the high density means there is a lot of protein, a little bit of fat, that's high density lipoprotein, HDL, that's good. But if you have LDL, that means you have low protein but high fat. That's dangerous, LDL. Are we clear? Yes, so now if you have protein with a lot of cholesterol in the center, then it's going to stick up to the blood vessels in the coronary circulation and clog them up. And that's going to cause the heart attack. Okay. Enzymes are proteins that function as biological catalysts. They are proteins that function as biological catalysts. So that's it. Are enzymes proteins or no? Are enzymes protein or no? They are proteins. Thank you. Some are ribozymes, composed of RNA and found in the ribosomes. So enzymes act on one or more substrates. So a substrate is the reactant, like a customer. Substrate for, so now you are here, a nursing student. If you go in the airplane, nobody's gonna call you nursing student, they're gonna call you a customer. When you leave, you're no longer a customer. Same thing. Reactants are reactants, but once they are inside the airplane, once they are inside the enzyme, they're now a substrate, because enzyme deal with substrate. We need to convert them into reactants. So they speed up the chemical reaction by lowering the activation energy. So the energy needed, and that is the energy needed to get a reaction started. So it permits the reactions to occur rapidly at body temperature. So they speed up the reaction. Enzyme naming convention is named for the substrate with ACE. So if there's an ACE, that's an enzyme. So when we did the sugars, who breaks down sucrose? It's sucrase. Who breaks down maltose? It's maltase. Who breaks down lactose? It's lactase. What does the enzyme maltase break down maltose into? The sugar maltose is made up of two monomers, and we split them in half so they can be shuttled into glycolysis. Who are the two monosaccharides that give me the disaccharide maltose? I drew them up here last week. 2 with 2, 2, 2. Yes. Galactose. Galactose is for milk, lactose. It's not for maltose. I told you the name is easy. Galactose is for lactose. And I told you the first point is that the three of them have glucose already. So now you already know 1 out of 2 for the three of them. Sucrose, maltose, and lactose. All of them are made up from 2, 2, 2. And each one of the two is glucose, glucose, glucose. Now you need to worry about the second one. And the easiest one I told you is lactose, because it's galactose. So it has glucose and galactose to give you lactose. Yeah, 2 glucose together give you maltose. And who gives me sucrose? Glucose and fructose. So now when I want to break them down, I use enzymes, proteins. So I use the enzyme sucrase to break down sucrose into glucose on one side, fructose on the other side. I use lactase to break down lactose into glucose and galactose. And I use down maltase to break down maltose into glucose and glucose. So that was a protein. You see, that was an enzyme. The one that cleave things together, that's an enzyme. So the enzymes act on one or more substrates. Okay, speed up the chemical reaction by lowering the activation energy. So the energy needed to get a reaction started. It permits the reactions to occur rapidly at body temperature. So the enzyme naming convention is named for the substrate with A's as a suffix. So examples are amylase, and it catalyzes the hydrolysis of amylose, starch. Lactase catalyzes the hydrolysis of lactose, milk sugar. Oh, there we see, you see the activation energy, we have dropped it. We have dropped it. So that was the activation energy. I need to climb. I need to climb this activation. That's why they call it activation energy barrier, the yellow. You see how big it was? But now I have dropped it. You see how little it is? So now the enzyme, you see that is reaction occurring slowly without a catalyst. And that is a reaction occurring quickly with a catalyst. Are we clear? Do we now appreciate? So if I show you a picture like that on the exam without all the words, you can see that that's the free energy that we need to have. And as time goes on, what's the difference between the two graphs? This one had a big mountain I gotta go up to so I can get here, and this one had just a little bump. So that means the activation energy, the mountain peak dropped. It was Mount Everest. I gotta go all the way up. Now it's just a little hill. It's just a little hill that I need to get over. Enzyme action. So substrate binds to pockets on the enzyme called the active site. An enzyme called the active site. So the formation of the enzymes, there's a formation of enzyme substrate complex. Enzyme substrate complex. So the enzyme substrate, there's an enzyme substrate specificity is like a lock and a key. So the enzyme binds to the substrate so tightly like a lock and a key formation. So the enzyme releases the reaction products and the enzyme is unchanged and can repeat the process. So these are the really important things we need to know about the enzyme. Some people think that the enzyme who does this job, can you please look here? You see this graph? That shows you what the enzyme can do. What can the enzyme do? So what does the enzyme do? The enzyme, that is the free energy, and that is the time. So without an enzyme, I gotta go up Mount Everest, come down. With the enzyme, I gotta go up to a shorter distance. Maybe I need to go up like that and come down. So here, the activation energy, EA, was so high. And here, the activation energy with the enzyme is so low. So that's how we speed up the reaction. So now what happens to the enzyme when the reaction is over? Is it destroyed? It's gone? No, the enzyme will never be consumed in the reaction. It just speeds you. Like when the subway gets you faster from your home to here, is the subway going to be disappearing after it does that? It's always there. It's ready to take somebody else now. That's the first thing. Enzymes are never consumed in the reaction. Second one, if you tell the enzyme, get me from A to B, is it going to take you from A to C or A to D or A to E? Is it going to take you somewhere else? If this reactant gives me this product, if I use an enzyme who makes it faster, is it going to give me something else? No. So that's an example of an enzyme specificity. It will never change anything. It just makes it faster. But it's like the subway, the cab driver. He gonna get you to your school. It's not like when you get on, it's gonna take you somewhere else. It's just gonna get you here faster. That's exactly what the enzyme does. It gets you where you need to go faster. Are we clear? So they are never consumed. They don't change the reaction. They just make the reaction faster by lowering the activation energy. That is really important. So the substrate binds to a pocket on the enzyme called the active site, and the formation of an enzyme, there's a formation of an enzyme substrate complex, and the enzyme substrate specificity is like a lock and a key, and the enzyme releases the reaction product. So the enzyme is unchanged and can repeat the process. So for example, the substrate sucrose is hydrolyzed by sucrase into the reaction products, glucose and fructose, glucose and fructose. There we go. That's our enzyme. These are our substrates. So the enzyme will take the substrates. Remember, I told you every figure, you got to know it well. Then it's going to take the enzyme substrate, then it's going to break this bond, and then it's going to separate them. So what's that an example of? Dehydration or hydrolysis? Here, when I added water, so what did I do here? They were connected. Green was married to blue. Green was married to orange. Then I used the enzyme, and now they are separated. So I used water. Did I have plus water or minus water? Plus water. So when I break down, you see the water, OH went here and H went there. What is that an example of? Hydrolysis. Thank you, thank you, thank you, thank you, and thank you. So we talked about how the temperature and the pH and other factors can change the enzyme shape and function. And they can alter the stability of the enzyme to bind the substrate. So enzymes vary. They vary in optimum pH. And the enzymes vary in the optimum pH. So the salivary amylase works best at pH 7. And the pepsin works best at pH 2. Even though both of them are proteins. But one works better in your mouth, in your saliva, that's pH 2, and the other one works best at pH 7. So the temperature optimum for the human enzyme is usually near the body temperature. Is usually near the body temperature. So many human enzymes require a non-protein partner called a cofactor. Many human enzymes require a non-protein partner called a cofactor. So the cofactor may be inorganic or organic. And inorganic cofactors include iron, copper, zinc, magnesium, and calcium. And some of these work by binding to the enzyme, triggering a conformational change that activates the active site. So organic cofactors are called coenzymes. So the inorganic ones, they are like metals, but the organic cofactors are called coenzymes. So they are often derived from vitamins. So for example, NAD plus derived from niacin acts as an electron shuttle between the metabolic pathways, glycolysis, and aerobic respiration. So you see in glycolysis, NAD plus gains an electron and it goes and supply to the aerobic respiration. In glycolysis, we get pyruvate from glucose, and in aerobic respiration, we convert pyruvate into carbon dioxide and water. And that gives us our ATP. That gives us our ATP. Look at glycolysis and look at aerobic respiration. In glycolysis, we had our glucose, and we ended up with pyruvate. And aerobic respiration will say, thank you, glycolysis, for giving me pyruvate. I'll take pyruvate and convert it into carbon dioxide and water. And that's how I end up with a buildup of metabolic waste of carbon dioxide, because I've converted pyruvate into water. And now the electron bind to NAD plus, and they donate it in the aerobic respiration. So converting glucose into pyruvate gives me energy. I lose electrons, so that's oxidation. That electron is going to be picked up by NAD plus and shuttled into the aerobic respiration pathway, and that's gonna add the electron into the pyruvate pathway, and that's gonna give us ATP. That's gonna give us ATP. So metabolic pathways. A metabolic pathway is a chain of reactions, each catalyzed by a different enzyme. So a simple metabolic pathway may be symbolized as follows. A metabolic pathway is a chain of reactions, each catalyzed by a different enzyme. So a simple metabolic pathway may be symbolized as follows: A to B, B to C, C to D. And A is the initial reactant, and B and C are intermediates, and D is the end product. The Greek letters are alpha, beta, and gamma, and the above reaction arrows represent the enzymes that catalyze each step. So A is the substrate for enzyme alpha, B is the substrate for enzyme beta, C is the substrate for enzyme gamma. So the metabolic pathways are turned on or off by altering the enzyme activity. This may be achieved many ways. This may be achieved by many ways, including binding or dissociation of cofactors, and the end product inhibits an enzyme at an earlier step, where D binds the enzyme and shuts down the production of intermediate product B. Pathway is turned on when end product is in demand. ATP, other nucleotides, and nucleic acid. Nucleotides are organic compounds with three components: nitrogenous base, sugar. So the nitrogenous base is a single or double carbon nitrogen ring, and the sugar is a monosaccharide. There's one or more phosphate groups. So the nucleotides are organic compounds with three components. So there's a nitrogenous base. So now we're talking about nucleic acid. So now we're done with proteins. Yay, we finished proteins, we finished fatty acids, we finished sugars. Now the only one that's left is nucleic acid. Yes, thank God. Let's do that now. Let's do now the nucleic acids. Now, how do the nucleic acids look like? The nucleic acids are made up of, you see, here we're talking generally. I'm never going to give you a structure and tell you, is that fructose or is that galactose or is that, I'm not going to show you something like this. Please look up there. Please look up there. Here, that's lactose. That's maltose. That's sucrose. And these are the single guys. That's glucose, that's fructose, that's galactose, yellow, glucose, green, fructose, brown. Am I going to ask you one example? Am I going to bring the yellow one and say, what is that? Is that galactose, glucose, or fructose? Am I going to do that? No. Am I going to bring the two and say, green and orange? Does that give you sucrose, or is that lactose or maltose? Are two greens give you mu? I'm not going to ask you a structure like that. But am I going to show one of those, green, yellow, or brown, and tell you, is that a fat? Is that a lipid? Is that a sugar? Is that a peptide? Am I going to do that? Obviously, because that's like, I have somebody who doesn't know what a monkey is. And I bring them the chimpanzee, the orangutan, and the different species of monkey and tell them which one is that monkey. That's too detailed. Okay, all I need to know what a monkey looks like, that's the one I gotta know that one, what a monkey looks like. But I don't need to know if it's an orangutan or is it a chimpanzee. Same thing you. You don't need to know what lactose exactly look like and what glucose and fructose exactly look like. I'm never gonna hold you that. But generally, you gotta know what it look like from outside. Don't need to know specific, but you gotta know what it look like. Same thing, amino acids. I'm not gonna ask you, is this tyrosine? I'm not gonna ask you that. Like this is a lipid. You got to know that's the glycerol, and these are free fatty acids. Am I going to say this one on the top, this one in the middle, is this stearic acid or palmitic acid or linoleic acid? Am I going to do that? I will never do that. But you need to know generally that this is a glycerol. That's not a sugar. That is not a sugar. That's a glycerol. That is not a sugar. That's a free fatty acid, because these are the guys who give me my OHs. Okay? Here, that's what I was talking about earlier, the phospholipid. Remember, it has a phospholipid head. It has a choline. You see the choline? There's a choline phospholipid head. And then the rest is a tail. You see the tail is hydrophobic and the head is hydrophilic. So that's cholesterol 4 rings. And look, these are the amino acids. Am I going to tell you this one? Is this methionine or cysteine or tyrosine or arginine? No. But you need to know that this structure with a carbon, 1H here, carbonyl carbon, carboxyl group, an amino group, and an R group, then I get that structure. Okay? So you need to know the general structure, not the name, the general. Same thing with, now let's talk about what is a nucleotide look like. Please look up here. Nucleotides look like this. They have a nitrogenous base. Nitrogenous base. Then we have 4 different nitrogenous bases. Adenine, thymine, cytosine, and guanine. Adenine binds with thymine with 2 hydrogen bonds, and guanine binds with cytosine with 3 hydrogen bonds. But these are called ATGC, are called nitrogenous bases, and they are divided into groups. If you have 2 rings, you are called epurine, and if you have 1 ring, you are called pyrimidine. Are we good? So, pure or good? Pure or good, meaning that purines with 2 rings are your adenine and guanine. These are your purines, adenine and guanine. They have 2 rings, and cytosine and thymine, they have 1 ring. But they don't bind with each other, they bind with these guys. The A binds with the T with two bonds, and the G binds with the C with 3 bonds. But in the end, what you need to know for now is that these are nitrogenous bases. So every nucleotide is called a nitrogenous base. So I can have any random nitrogenous base. It could be A, it could be, what are the other 3? A, T, C, G. In addition to the nitrogenous base, I gotta have a sugar, a ribose sugar, a ribose sugar. Say a sugar, just say a sugar. We need to have a sugar. And besides the sugar, I gotta have a phosphate. Now, what does ATP look like? ATP is adenine. You have adenine, who is associated with a sugar, who is associated with my black nitrogenous base is adenine. Adenine, the sugar, and the phosphates. I have how many? 1, 2, 3 phosphates. This is known as adenine triphosphate, ATP, adenine triphosphate. When I break this bond, catabolism, endothermic reaction, exergonic reaction, I'm going to end up with ADP. And then I break down ADP, I break down 1 more bond, and that gives you AMP. Okay? You see? ATP, ADP, AMP.
The ATP, when it breaks.
ATP when it has 3. It's adenine triphosphate. That's ATP. I lose 1, I have 2 now. That's ADP. I lose 1, I have 1 now. That's AMP, monophosphate. Okay? Okay, guys? That is what a nucleotide look like. Now, what if I remove all the phosphates? Remember in my nucleotide formula, what does the law say? What does the law tell me? The law says if I gotta be a nucleotide, I gotta have all 3. A nitrogenous base, A, T, C, or G, a sugar, and at least 1 phosphate. That makes me a nucleotide. That makes me a nucleotide. But what if I lost 1 phosphate? So now I'm just nitrogenous base and a sugar. No phosphate. What does that make me? Am I a nucleotide now? I'm a nucleoside. I'm a nucleoside, not a nucleotide. Nucleoside. So now we went down. We went from the president to vice president now. So now we are not nucleotide. We are nucleoside. We are not a general anymore. We are a lieutenant. We dropped one level down. So the DNA is a bond between the nucleotides of one side, and there will be a bond on the other side. So, okay, we know that nucleotides are adenine, sugar, and a phosphate. Adenine, sugar, and a phosphate. So that's one strand. On the other side of my DNA, I have a nitrogenous base, and I have another nitrogenous base. Then you have a sugar, a phosphate, okay, so these are my two. Are you guys paying attention? You're never gonna see this, so please pay attention. You're never gonna. I'm not gonna. You're gonna see it, but you're not gonna have somebody who will break it down. Please pay attention. These are my two strands. That's strand number one. Strand number one. That's strand number two. For strand number one, the nucleotides are connected by the phosphate groups. So they're connected vertically to make a long line skyscraper going up by this vertical bond known as, is between two phosphates, known as phosphodiester bond. So the bond between the amino acid is called peptide bond. The bond between the sugar is called glycosidic bond. The bond between the nucleotide is called phosphodiester bond. Over there on strand number two, you have here a phosphodiester bond. Same thing. You have a nitrogenous base, a sugar, and a phosphate, and they're all connected by phosphodiester bond. They are all connected by phosphodiester bond, sugar and adenine, or maybe some form of nitrogenous base. So now they connect vertically with what kind of bond? They connect vertically with what kind of bond? Phosphodiester bond. Phosphodiester bond. Phosphodiester bond. Phosphodiester bond. They connect vertically. Now, horizontally, we said that they connect if it's, let's say the nitrogenous base here is a G, and G binds with C, and T binds with A, and C bind with G. We said that A and T bind with how many bonds? 2 bonds. And G and C bind with how many bonds? 3 bonds. C and G bind with 2 bonds. So that shows you the bond that is holding the strand up is phosphodiester, really strong bond. And the bond that makes them connect with each other is a hydrogen bond. That's a hydrogen bond. That's a hydrogen bond. So they stick together. That's why when you heat it up, they don't fall apart. They fall away from each other because heat breaks which bond? Heat breaks phosphodiester bond or hydrogen bond. It breaks hydrogen bond. So it breaks this bond right here. It breaks this bond right here. Okay? Okay, now let's go to our amino acids. There we go. We talked about that. You see, nucleotides are organic compounds with three components: a nitrogenous base. Nitrogenous base is single or a double carbon nitrogen ring. It has a nitrogenous base. We said one has two ring and the other one has one ring. The sugar is a monosaccharide and one or more phosphate groups. We said that, right? They made up from nitrogenous base, sugar, and phosphate group. Example of ATP. ATP is adenosine triphosphate. Has adenine, nitrogenous base, ribose sugar, and three phosphate groups. So you have adenine, ribose, and triphosphate. Adenine with two rings, ribose with one ring, and triphosphate with three rings. Adenine triphosphate is the body's most important energy transfer molecule. So do you guys see what I was telling you? Nitrogenous base, a sugar, a ribose sugar, and a triphosphate. So every nucleotide must have the three: the nitrogenous base, a ribose sugar, and triphosphate. So adenosine triphosphate, ATP, is the body's most important energy transfer molecule. It is the body's most important energy transfer molecule. So it stores energy gained from exergonic reactions. So it releases it within seconds for physiological work. So it holds energy in covalent bonds between the phosphates. Second and third phosphate groups have high energy bonds. Most energy transfers to and from ATP involve adding or removing a third phosphate group. Hydrolysis of ATP is catalyzed by adenosine triphosphate. You see, when we hydrolyze ATP by adenosine triphosphatase. Any questions? Yeah. Somebody have a question? It breaks the third high energy phosphate bond of the ATP to produce adenosine diphosphate, inorganic phosphate, and energy. It releases 7.3 kilocalories of the energy per molecule. There's also phosphorylation. Phosphorylation is the addition of free phosphate. Addition of free phosphate group released from ATP hydrolysis to another molecule to activate it. It is carried out by the enzymes called kinases. So you see the source and uses of ATP. Glucose are converted into which release energy, which is used for, as you see here, which is then converted into muscle contraction, ciliary beating, and active transport and synthesis. So the energy of ATP synthesis comes primarily from glucose oxidation. The first step is the first stage is glycolysis, which is splitting of glucose into two pyruvate molecules. A little ATP is made in that process, and most chemical energy remains in pyruvate. The fate of pyruvate depends on oxygen supply. So if ATP demand outpaces oxygen supply, pyruvate undergoes anaerobic fermentation to lactate. If enough oxygen is available, aerobic respiration occurs in the mitochondria. Aerobic respiration occurs in the mitochondria. So that shows you glycolysis, and that is anaerobic respiration to give us lactate. And this is the oxygen where you have aerobic respiration. It requires oxygen. Okay. Other nucleotides. So guanosine triphosphate is another nucleotide involved in energy transfer. In some reaction, donates a phosphate group. So cyclic adenosine monophosphate is formed by the removal of second and third phosphate groups from ATP. So in many cases, its formation is triggered by a chemical signal. binding to a cell surface. So cyclic AMP becomes second messenger within the cell. See that cyclic that's CAMP. It is cyclicized and the monophosphate is attached. The phosphate is attached to the ribose sugar. Nucleic acid, we talked about those. The DNA, deoxyribonucleic acid. So what's the difference between DNA and RNA? DNA sugar, the sugar of DNA here, you see this ribose sugar? The only difference between DNA and RNA is that why they call DNA deoxyribose, because it has no oxygen here. The oxygen is gone. But the other one is called a ribose. That's why we call this a ribose sugar. What's the name? You see this sugar right here? It's called ribose because it has oxygen on the second position. But in the DNA, that oxygen is gone. It's devoided. That's why they say deoxy. So the DNA is deoxyribonucleic acid because the sugar that it has no oxygen on the second position. But the RNA is ribonucleic. It doesn't have a missing oxygen. That's why we don't call it deoxy. We call it oxygenate. Okay. All right. So we are done with right? We are done now with our macromolecules. We're done with fats, how they look like, how they bond together. We are done with sugars, what they are, how they bond together. Done with proteins, what they are, how they bond together, and this nucleic acid, how they bond together with phosphodiester bond and your hydrogen bonding. So now let us now move on to let us now move on to part 2.